Hey guys, welcome back to another lecture video for Chem 104. I forgot to talk about cellulose in the previous lecture video, and so I'm just going to go ahead and talk about it here. So cellulose is our last polysaccharide. And so this specific polysaccharide has a structural role in plants. And so cellulose is typically found in the cell walls of plants. And so these cell walls provide the structure that plants need, kind of like a backbone to a human. And so cellulose is composed of glucose, of many glucose units. And so it turns out that, among the common polysaccharides that we've discussed, amylose, amylopectin, glycogen, and cellulose, all of these common polysaccharides are composed of numerous glucose units. And so glucose feels like a very diverse carbohydrate with many different functions. And the function is going to be dictated by how those glucose units are kind of bonded to each other. And so anyways, cellulose is an unbranched polysaccharide. So that's very similar to amylose. However, cellulose is not similar to amylose because the bonding for cellulose is beta 1,4, whereas for amylose, it's alpha 1,4. And so just the flipping of that hydroxyl group on the anomeric carbon, beta means that the hydroxyl group is going to be found on top of the plane of the anomeric carbon. Whereas alpha means that the hydroxyl group is going to be found at the bottom part of the anomeric carbon. And so overall, cellulose is more resistant to hydrolysis than starches, and cellulose cannot be digested by humans. The reason why is because we do not have an enzyme that could recognize and that can break a beta 1,4 glycosidic bond. The only enzyme that we have in our body that we naturally produce is an alpha 1,4 glycosidic bond. If we wanted to be able to digest cellulose and use cellulose as a source of energy, then we need our bodies to produce a beta 1,4 glycosidic bond. And so organisms that utilize cellulose as a source of energy will have an enzyme that can recognize the beta 1,4 and break it and hydrolyze it. And so looking at this this image here, all of this is the cell wall. Okay. And so if we kind of zoom in that cell wall, we get these cellulose fibers. You can kind of see that it's layered in such a way that they're crossing each other. It's kind of like a braid, but not as neat as we would do braids as humans. Anyways, I want to go ahead and zoom in on a couple of these glucose monomers, just so that you guys can understand the beta 1,4 linkage and its consequence with respect to cellulose. And so I'm going to go ahead and look at this glucose monomer on the left. So this is our oxygen, that's our reference point. So we know that this is going to be our anomeric carbon. So this is carbon number 2, carbon number 3, carbon number 4. So in carbon number 4, if you have this molecule right side up, that means that the OH the hydroxyl group on carbon number 4 is pointing at the bottom. Okay. And so for this to be beta, that means that the hydroxyl group has to point up. And so I want you guys to notice that in this cartoon image of cellulose, the glycosidic bond is pointing up. And the only way that's possible, actually, let me retract here. Actually, no, I'm going to keep going. So the only way that's possible is if carbon number 1 and carbon number 4 have hydroxyl groups that are both pointing up. So in other words, they're both parallel to each other. But I just said a little bit earlier that on carbon number 4 of glucose molecules, that these hydroxyl groups are typically at the bottom. And so what that means is that for a beta 1,4 glycosidic bond to form, this second molecule, the second glucose molecule, has to be flipped upside down. Okay. And by flipping the second glucose molecule upside down, this will allow this hydroxyl group that is typically found at the bottom to now be on top. And so this is observed by looking at other reference points. So I want you guys to notice, I'll do this in pink, I want you guys to notice that the oxygen in the ring is found more to the back, according to the Haworth structure, right? However, if you guys look at the second glucose molecule, notice that the oxygen that's in that ring is now facing the front. And so if you're going from back to front, then that means you flip the molecule. And so the carbon number 4 here, by flipping it, will have a hydroxyl group that's now parallel to the hydroxyl group that's on this anomeric carbon. And so once your hydroxyl groups are aligned, then a dehydration reaction can occur. And that dehydration reaction will form a glycosidic bond such that it's pointing up. And so for the second molecule, if you flip the second molecule so that carbon number 4 is pointing up, that means that for the second molecule, the hydroxyl group that is on the anomeric carbon is going to point down. However, all we did was we flipped a beta, right? And so since we flipped a beta, this is still technically beta because it was originally on the top. All we did was we took the molecule and we flipped it upside down, right? So it's still the same molecule. It's still a beta glucose. But the consequence for us flipping that molecule so that carbon number 1 and carbon number 4 aligns would also flip the position of the hydroxyl group that's on the anomeric carbon. And so if you look at the next glucose molecule, the oxygen is now found recessed at the back. And so this is the third glucose molecule is not flipped. And since it's not flipped, then that means on carbon number 4, the OH is naturally pointing down. And so since the anomeric carbon and carbon number 1 that was flipped is now in line with the hydroxyl group that's on carbon number 4, these two can come together to form a glycosidic bond when a dehydration reaction occurs. And so overall, if I just kind of zoom out and just look at the trend, we have a glucose molecule that's right side up, and then this one's upside down. right side up, upside down. And so the beta 1,4 glycosidic bond conforms a very specific spatial arrangement of these glucose monomers, such that in a more global level, the ability for, not so much the ability, but because the glucose monomers are right side up and then flipped, right side up and flipped, right side up and flipped, it allows the hydroxyl groups, it allows the hydroxyl groups among different strands of cellulose to interact together, forming a hydrogen bond. So all of these dots that you guys are seeing as I zoom out, they're all representing hydrogen bonds. And as we know, hydrogen bonds are very strong intermolecular forces. And with, hundreds of thousands of glucose units, all having these hydrogen bonds, we have hundreds and thousands of hydrogen bonds that is keeping these strands held together. Okay, and so if you guys recall glycogen and amylopectin, all of those glucose monomers were either was either linear or it was branched. And so for cellulose, for those glucose, those strands to kind of interact together, they're not held by covalent bonds through a branching point. What they are held by is simply an intermolecular force, which is a hydrogen bond. And since these hydrogen bonds are very strong intermolecular forces, then it's enough for these individual strands of cellulose to form microfibrils, to later then form cellulose fibers. And the cross weaving of these cellulose fibers, if you will, is enough to create a very rigid cell wall. Okay, that will enable the cell to, keep its shape. And so overall, if your plant cells are plump, in other words, are like, they have enough water, it's going to form a very rigid structure. So it's crazy to think that, one little thing, such as alpha and beta, they have such a tremendous global effect that dictates whether or not that specific polysaccharide can be used as a source of energy, or two, it can be used for structural, to support, like, for example, the creation of cell wall. And it's only from, this idea of alpha and beta. So for beta 1,4 glycosidic bond to occur, those two glucose monomers, they have to be flipped with respect to each other. Like one of them is right side up, the other one's upside down. Okay. And that consecutive pattern, once again, enables these individual strands to form hydrogen bonds among themselves, which is a strong enough intermolecular force to create these cellulose fibers without the use of a covalent bond. All right, so that's pretty much it for cellulose. What you guys just want to do with the polysaccharide section is to compare and contrast their features, what's similar about them, what's different, and primarily, what they're used for.